I'd say we taste a little like chicken. I just realized that I got this all set up, but the one thing I did not do is turn on the microphone and the receiver for the microphone. So I've been talking, and you haven't been able to understand anything I've said. So we're going to talk about nutrition and digestion. Chapter 14. The old disappearing mouse trick. Not too surprisingly, there's a lot of cartoons that have to do with feeding. Now, there are some animals that are in a situation where, like a tapeworm, they live in a digestive system, so they just need to absorb the materials. It's a somewhat uncommon way of feeding, but it does happen. We've talked about filter feeding, also sometimes known as suspension feeding, where you are pulling particles out of the water, straining them out. Personally, I've always felt that weaver spiders should be considered filter feeders because they are filtering insects out of the air with their webs, but nobody ever applies it to air. They always apply filter feeding to water. Deposit feeding. Scraping food off of surfaces. Snails do this. Very often, working your way through a medium and eating, the way the earthworms consume the dirt that they're working through. And a lot of things do that with aquatic sediments. That is very commonly included in deposit feeding. Animals eat fluids. Aphids pull fluids out of plants. Mosquitoes primarily pull fluids out of plants. Is that the females draw blood because has necessary nutrients for producing eggs. The males never do that. They're plant fluid eaters. But plantable and plant or animal fluids. So very often involves some sort of injectable feeding apparatus and then something to suck the fluids through. And then you get massed food, which processes three different ways that aren't necessarily always separate, often included. It may be taking food in as in its entirety, like the way that a snake eats, right? Snake catches an animal, swallows the whole animal whole. Is that a lot of animals that eat insects will grab the insect and swallow the insect whole. It may involve taking food and biting it into big pieces and swallowing those whole. And it may involve taking food and grinding it up into little bitty pieces and swallowing that. Those are all various ways of massed food consumption. One of the interesting things about mammals, I think I mentioned, maybe I didn't mention because I didn't really go that much through the groups of vertebrates, is that one thing that makes us different from the other vertebrates is that we have multi-purpose teeth all in the same mouth, whereas reptiles tend to have teeth that are more or less the same purpose. Some of them are grinding. They may be different sizes and stuff, but they tend to have a bunch of grinding teeth. Some of them are tearing teeth that you'd have in predators. They may be different sizes, but they all tend to be the same sort of tearing teeth. We have chopping teeth, we have tearing teeth, we have grinding teeth all in the same mouths. It gives us much more niche flexibility. Our digestion is linked to digestive enzymes, proteins that attach to large molecules and help them to fall apart into component molecules. You tend to think acid, right? Acid is not really digestive. The main purpose of acid is to take those big molecules and get them to unwind so that the excuse me, the enzymes can then get at them. But the acid itself doesn't break the molecules down. It loosens the internal connections of a big blobby protein and makes it more of a long stringy protein that then can be snipped apart into its components. Some animals, intracellular, I mean protozoa for sure, pull stuff in, digest it in food vacuoles. Cnidaria and sponges do that to some extent, where they pull in particles into the cells individually. Cnidaria is a kind of a combination of extracellular in the digestive cavity, but when particles get small enough, even though they're not completely broken down for use, they may take the big things into cells individually and break them the rest of the way down there. One of the things that makes animals is an internal digestive space. His food is broken down and absorbed from an internal space. That space is extracellular. It's actually technically outside the body, even though it's inside the body. I think you know what I mean. So you have a digestive cavity with specialized lining, usually some sort of protection to keep the lining from being digested itself. That's very commonly mucus, but there's other things that go on the linings as well. Cnidaria and platyhelminths, flatworms, have an opening the food goes in through, and it gets processed and broken down. Pieces are absorbed. Whatever is not breakable or isn't going to be absorbed is then released back out through the opening it came in through. It's a sac, a single opening sac. Is that starting with the roundworms, you wind up with an entrance, a tube, and an exit. And there's a couple of huge advantages to that. A sac, you got to put the food in, then you got to break it down in stages. You can't be putting more food in while you're working. If you're midway point of digestion, you can't be adding more food to the mix. So a tube, you can just put it in the front end, and it gets processed in stages along the tube. The tube can specialize for that. That's another advantage. And also you can keep putting food in the front end and have it continue to be broken down in stages. So the tube is definitely a huge advantage over the sac system. So we're going to pick up that idea of the sac system. Oh, sorry. In small systems, cilia can produce enough of a current to move stuff along. Somewhat unusual, but you do find it. In some systems, there's a lot of mixing that goes on. This is what happens in our stomachs. It's an old term for segmentation, but it's often applied. And it's churning and mixing the juices together with the food. In most tube systems, there's a traveling squeeze that moves the food along. Peristalsis, like squeezing a toothpaste tube. It's what moves annelids, a lot of annelids along, but it also is what moves food through their digestive system, because they do have a tube digestive system. Once people realize that we have a microbiome that helps us a bunch of different ways, including digestive, but also for a lot of other things, then they started to look elsewhere. In pretty much every digestive system you look closely at, there's always microbes living in the digestive system. And if you look closely enough, at least a decent number of them are there in some sort of trade-off, where they not only are benefiting from a good place to live with stuff that they can live off of. Our symbionts often live off of stuff we can't actually digest. They recycle mucus. Some of them go after certain types of carbohydrates we can't break down. But also they give back. They're symbionts. We give something to them, they give something to us. And that's becoming a very, very common discovery in animals from Cnidaria all the way up, to us. Although I shouldn't really be talking about us up in that type of context. There's regional breakdown for the tube. At the front end, you have the receiving region. Very often there are specialized mouth parts involved. Could be just biting, could be chewing, could be storage in some cases. What they call a buccal cavity, like you'd see a chipmunk with its cheeks full of food. Not unusual for there to be salivary glands. Saliva is, in animals that chew food up into small bits, the saliva allows you to temporarily glue that together in order to swallow it. We talked about that in the rat lab. But also some saliva has digestive qualities. Got to be careful about what you digest in saliva, because it's hard to protect the lining of the receiving region from digestive enzymes. So you don't want to go after something that your tissues are made of, like proteins and lipids. And generally the swallowing region, in us our pharynx, is the very end of the receiving region. And then you're into you could put the buccal cavity in here. You could put the pharynx in here. There's a lot of overlap on these various regions. But your esophagus, your swallowing tube, is definitely a part of the conduction region. A lot of animals have crops. They put the food into a big storage device. They bite a bunch of stuff off, put it in the crop, go elsewhere where they're more protected, and then let their system work the food. I mean, it's basically what's happening with a chipmunk and its buccal cavity. It's pulling out a bunch of food and then going and hiding somewhere where it's going to process it. Oops. Grinding and predigestion. Now we grind in our receiving region, but an awful lot of animals have their grinders farther down, like a gizzard, birds, a whole bunch of insects. They swallow fairly large bits, but they are going to grind it up before it gets into the final digestive area. You want it in small digestible bits. Predigestion, I talked about acid, is that acid chambers, not super common in animals across the board, but acid is very useful at what's called denaturing large organic molecules, getting them to unwind. What we have in our stomach is acid and one digestive enzyme that is good at clipping a particular amino acid to amino acid connection. So you get this big blobby protein, you unwind it, the enzyme goes snip, snip, snip. You wind up with long strands of amino acids. You can't absorb that. It isn't really digestion, but it's got it. One of the problems with unwinding stuff with acid is that most of our digestive enzymes won't work in the acid. It's got to go into a chamber at more of a basic pH. And some proteins will unwind and hit the base and go zoop and go right back into a very hard to digest blob. Whereas if we've snipped them into bits, even big bits, they're going to be much more accessible. Again, this doesn't always happen here. Sometimes it happens farther down. Fermentation is a lot of plants. The fibrous material in plants is very difficult for animals to digest. There are very few animals that purely with their own enzymes can break down the heavy-duty plant fibers. But a lot of animals have chambers full of symbiotic bacteria that are capable of doing that. Or in some cases, in terms of wood eating, symbiotic protozoans that break down the lignin and the tough stuff that's in wood. But you can actually kill termites in the lab. You put them in a pressure chamber and you pump in oxygen at pressure. It pushes oxygen into the spaces of their digestive system. Their symbionts are anaerobes. Oxygen will kill them. And once they are dead, the termites will keep eating and swallowing wood, but they got nothing to digest it with. They will starve to death. Obviously, this is just something that somebody tested. Not exactly, the Orkin man is not coming along and killing your termites that way. And sometimes like deer and other related animals have a multi-chamber fermentation system. And sometimes they, like cows, they have a fermentation system. They work it a little bit. They bring it up. They chew on it a little bit more. And they take it back down and ferment it a little bit more. Terminal as in final. So this is where you're breaking down the food molecules into small absorbable components that you can then build your own molecules out of. So amino acids, parts of lipids, small carbohydrates. I mean, things like vitamins and minerals and stuff, you're not really digesting. You're just absorbing them across a pretty good surface for passive and active transport. The enzymes are generally in the space, some of them produced by the lining, some of them produced by digestive glands, in our case by our pancreas and liver, sort of. Your liver producing an emulsifier rather than a digestive juice. Still kind of need it. And a lot of animals have digestive glands, accessory organs, where the food doesn't go through them, but they supply materials for digestion. Very often, this is an area with increased surface area. The surface may be folded, a tube with folds on the inside. There's a lot of projections and microvilli. Tiny little, remember the tiny little cilia-like projections that expand the surface area of a cell. And then sometimes there's further processing where you've got an absorptive gland that is going to take the raw materials from digestion and fiddle with them, because some of them are potentially toxic. That's the main job that your liver does. Your liver takes some molecules that in your general circulation would be toxic, like ethyl alcohol or formaldehyde, and things that are found in like slightly, fruits and things that have been gone a little bit over, but are still edible. And your liver processes that into more benign molecules, sometimes more usable molecules. Tricky thing if you're a drug developer to get stuff past the liver, and sometimes they make it work for us, is that the chemical in Tylenol that makes your headache go away is not the chemical in the pill. It's what your liver makes of the chemical in the pill. Okay, one more thing. I don't want to squeeze it in the bottom here. water absorption and waste concentration. Is that, sorry, digestion has to happen in a soup, a water-based soup, because the digestive enzymes have got to mix with everything, and then the components have got to be able to get to the linings and get absorbed. But water is an important part of our metabolism, and we're always losing it to the environment. We need to restore it. And so you get the water out late, farther down the system, when you're taking what you couldn't digest and concentrating it. In some cases, adding things from the body. In the case of humans, is a decent amount of stuff comes out of your liver that are metabolic wastes, breakdown products that your body doesn't really have a use for anymore, that are added to waste materials. When we recycle our hemoglobin, I think I mentioned this, is that there's a brown breakdown product that goes into your digestive system. Makes everything brown. And other sorts of things are concentrated. About somewhere around a quarter of what comes out the back end of most mammals is added from metabolic wastes. About a half of it is undigested food, components we can't break down and absorb, or haven't broken down and absorbed. And about a quarter of it is symbionts, is that those bacteria. You lose a lot of bacteria all the time. Good old feces. Hunger can be specific. In some animals, they are driven to find a particular nutrient. Is that in the Amazon, birds can fly a long distance and actually consume clay in order to get certain minerals that are not accessible through the fruits and leaves that they would normally eat. Rainforest, very often, it leaches a lot of this stuff away. And we're bad at that. Some people have suggested that that the cravings that women get when they're pregnant is kind of a holdover of a drive to get certain nutrients. There's no evidence that it really is that. It's not specific enough. But it may be a holdover from an earlier time where we were able to do that. And some animals very much can. Okay, I've talked about these. Carbohydrates, you need carbohydrates, you need proteins, you need lipids. You need minerals. Minerals are integrated into a lot of our proteins. Charged ions are a big part of our chemistry. Your muscles work using the movement of ions in and out of the cells. Your nerves work moving ions in and out of the cells. Vitamins are enzyme helpers. The term specifically means coenzymes that humans need to get from their food. We don't make them ourselves. It's not applied that way most times, but it really is a human-centered term where the list is particularly a human list of things that that we get from our food. Considering we've been omnivores for a really long time, there's a whole bunch of coenzymes on that list that other animals do make. Your cat makes vitamin C. Doesn't normally get it in its diet. But it needs to be able to have that coenzyme available, so it makes it. We don't. Our ancestors have been pulling it in for a long time. Okay, that's it for that particular chapter. Told you these chapters were going to go by fairly quickly. I know this is a somewhat decent length video, but it covers the whole chapter.